안녕하세요. 저는 호주 시드니 의대에서 해부학 교수로 있는 임경준입니다. 호주에 계시는 분들도 저기 한국의 교수님들처럼 연구와 수업을 병행하고 있습니다. 저는 인체해부학 수업을 하면서 cardiovascular research를 하고 있고 오늘의 발표는 저희 실험실의 연구 프로젝트 중 영양과 관련된 비만, 당뇨, 그리고 고혈압에 대해서 이야기해 보려고 합니다. 발표가 약간 지루할 수도 있지만 재미있게 들어주셨으면 합니다. 감사합니다. Thank you for the invitation to present our study at Chungang University. The topic of the presentation is on the diet-induced programming of adult disease, and in particular, I'll be focusing on the maternal obesity and the offspring health in later life. Now, I divided the presentation into two parts. First part is talking about the adulthood obesity. So in this part, I want to talk about the role of the brain in the hypertension, which is induced by obesity. In the second part, I want to talk about the programming of adult disease during pregnancy. So this is involving maternal diet and the maternal health. And the content will focus on the developmental programming and the prevention of the adulthood obesity-related hypertension. And I would also like to finish off with an alternative approach in treatment of obesity, diabetes, and also the pre-diabetes. So sit tight, and I hope you guys enjoy the presentation. Now, obesity is a popular topic, but to refresh our memories, let's go through some of the key changes and how they occur. So, obesity leads to increase in cytokines and free fatty acids, which causes insulin resistance and hyperinsulinemia, meaning there is an abundant amount of insulin in our body. There is an increase in plasma leptin, and this occurs as more fat means more production of leptin, and leptin is mainly produced by the fat or the adipose tissue. And there's also decrease in the ghrelin levels, which helps with generally leptin and ghrelin controls our appetite. So reduction in ghrelin leads to a change in the food consumption of the subject. There's also reduction in the bowel reflex. Now, all of these conditions eventually leads to increase in sympathetic nerve activity. Now, sympathetic nerve activity is 교감신경. In Korean, it's 교감신경. So there's an increase in the sympathetic nerve activity, which triggers the vasoconstriction, increased sodium retention, and also cardiac output, as well as changes in the structures of our body, causes the target organ damage, such as the damages in the blood vessels, the kidneys, as well as the heart, eventually leading to hypertension. And as we all know, hypertension is 고혈압. Now, there are a lot of evidence showing that the role of the sympathetic nerve activity is crucial in obesity-induced hypertension. Now, Murray Esler's group have pioneered in Australia with the renal noradrenaline spillover studies and have shown that in obese patients, which are in red, there are increase in the level of noradrenaline in the kidneys. Furthermore, Elizabeth Lambert's group also have shown that using the muscle sympathetic nerve activity recording, they also shown that the patients with obesity showed increase in the muscle sympathetic nerve activity. Importantly, obesity-related hypertension is neurogenic, meaning the brain is playing a key role in this development of hypertension in obesity. For instance, when a ganglion blocking agent, which inhibits or blocks the transmission between the neurons in the autonomic nervous system, was used, studies have shown that obese subjects show greater depressor effect, meaning the blood pressure went down when this was blocked. And these studies includes in human, rabbit, mouse, and rats. And as you can see, the red represents the changes in blood pressure in the obese subjects, and blue were in the control group. Now, in our laboratory, we have expertise in obesity research using the New Zealand white rabbits. Now, there are great benefits using the rabbit model in study of obesity, as when rabbits become obese with diet manipulation, they show similar physiological changes as seen in human obesity. So there are changes in the heart and also in the kidneys, as well as activation of renin-angiotensin system. There's increase in the plasma glucose level, triglyceride, and also insulin and leptin, as well as they show hypertension and increase in sympathetic nerve activity. Now, in our laboratory, we developed this diet program which shows that feeding a rabbit with a high-fat diet, which contains about 13% of fat, for just 3 weeks leads to increase in body weight, mean arterial pressure, which is the blood pressure, heart rate, and the level of norepinephrine. Importantly, we saw increase in the renal sympathetic nerve activity in the conscious animals, meaning 콩팥으로 가는 교감신경이 월등하게 증가를 한 거를 알 수가 있었습니다. Now, for those who are not familiar with how much fat is 13%, this is about the content of Big Mac from McDonald's. So imagine you ate Big Mac for 3 weeks, and this will clearly show changes in the blood pressure, body weight, and also the renal sympathetic nerve activity. Now, importantly, in our animal model, we have shown that this is clearly a neurogenic hypertension, meaning the brain is controlling this hypertension, as when a ganglion blockade was injected, there was a greater reduction in the blood pressure in the high-fat diet fed animals. Now, using our animal model and diet regime, using the 13% of fat, we have also shown that there is marked increase in plasma leptin, which is in red here, is our high-fat diet animals, and the blue are the control diet. And importantly, the visceral fat, blood pressure, and renal sympathetic nerve activity were all correlated by the increase in the levels of the fat. Now, noticing this consistent increase in plasma leptin and insulin in our animal model, and these hormones being the main appetite controlling hormones, we wanted to investigate what is the role of the leptin and insulin in the neurogenic hypertension. Now, in normal condition, during an anorexogenic phase, leptin and insulin enters the brain via the median eminence, passing the blood-brain barrier, and activates the pro-opiomelanocortin neurons, and at the same time, it blocks or inhibits the MPY and agouti-related peptide neurons. Now, once activated, this POMC neuron releases alpha melanocyte stimulating hormone, which is an agonist of the melanocortin pathway, travels down to a lower order neurons, such as the ventral medial hypothalamus, dorsal medial hypothalamus, and the paraventricular nucleus and the lateral nucleus. And at the same time, while this is happening, our body experiences reduction in appetite, an increase in energy expenditure, and all of these changes occur via the activation of sympathetic nerve activity. So, we investigated whether this pathway is altered or changed in obesity. Now, to refresh our memory, hypothalamus is the 시상하부 of the brain. 그러니까 두뇌의 시상하부에서 일어나는 변화가 어떻게 일어나는지에 대해서 연구를 했습니다. So, to test the role of leptin and insulin in the brain of obese animals, we used the rabbit model, and we blocked the receptor using the leptin and insulin receptor antagonists. And these were directly injected into the lateral ventricle using intraventricular injection, or ICV injection. And we looked at the changes in the blood pressure and the sympathetic nerve activity. Now, prior to these injections, the animals were either fed with a high-fat diet or normal diet for 3 weeks. And before this experiments were done in week 1 or week 3 to see whether the early changes due to the high-fat diet feeding is different to the later stage with the continuous of high-fat diet feeding. So prior to injection, they had this diet program, and they also received the surgical implantation of the renal nerve electrode. Now, this is to measure the sympathetic nerve activity in conscious animals. So the unique technique we have is measuring this blood pressure and sympathetic nerve activity in conscious animals, which provides more accurate and real-life physiological changes in the animal. So, as you know, rabbits tend to be relaxed in a confined and compressed area, as shown in the image here, which is our measuring box. And with a surgical implantation of the electrode to the renal nerve, we can retract the electrode and connect to a computer. And also by connecting a catheter to the ear artery, we could measure blood pressure and renal sympathetic nerve activity in real life. And these are the images of the measurement. So this is what we see in the screen when all the measuring devices are connected. And for the renal sympathetic nerve activity measurement, because we receive the raw data, we integrated the sympathetic nerve activity with the maximum response to smoke to the animals. So these are the integrated data of the renal sympathetic nerve activity, and also we have the data for the blood pressure. Now, before any drugs were given at the baseline data, this was consistent to our previous findings where when rabbits were fed with a high-fat diet with 1 week or 3 weeks, they showed marked increase in blood pressure, heart rate, as well as the renal sympathetic nerve activity. Now let's see what happens when these animals were receiving the leptin and insulin antagonists directly to the brain. Now, one of the most interesting findings in our early experiments were that the effects of leptin and insulin antagonist in the hypothalamus. When the insulin antagonist was given, there was a drop in blood pressure from 1 week and 3 week animals. However, there were no changes in the renal sympathetic nerve activity, indicating that the blood pressure control via the insulin is not derived by the renal sympathetic nerve activity. It could be a different sympathetic nerve activity, such as a cardiac sympathetic nerve activity or other major sympathetic nerve activity. Whereas when the leptin antagonist was given, interestingly, at 1 week of high-fat diet feeding, there was no change in the blood pressure or the renal sympathetic nerve activity. However, in the 3-week high-fat diet animals, blood pressure and sympathetic nerve activity was significantly reduced, indicating that there is a delayed action of leptin. So to leptin to kick in the brain to make changes in blood pressure and sympathetic nerve activity, it needs a long time of accumulation in our body. So with this study, we concluded that the insulin signaling pathway is altered, but this is not driven by the renal sympathetic nerve activity. Whereas the leptin signaling pathway is altered, but there is a delayed activation, and there is an increased response to leptin in the high-fat diet-fed obese animals. Now, after this, we wanted to see which part of the brain is actually driving this part, because with the ICV injection, or the intraventricular cerebral injection, the drug goes to everywhere, all the part of the hypothalamus. So we don't know which particular or which specific part of the brain is driving this change. So to answer which part is driving this, we focused on the ventromedial hypothalamus and the dorsomedial hypothalamus, which is the known part of the hypothalamus that drives the changes in the sympathetic nerve activity. So to investigate this, we performed a similar experiment where there was a direct injection to the dorsomedial and the ventromedial hypothalamus. So we developed this technique to target these areas, and we gave both part of the brain, so left and right side of the brain, with the drugs, and this time we used same drug, which is the leptin antagonist and the leptin antagonist, and alpha MSH, which is the agonist. So this is the same hormone when it's produced by the POMC neuron when leptin and insulin enters the brain. So we gave more of these to see what are the changes. And another drug is called the SHU9119, which is the blocking agent of this alpha MSH hormone. And also MPY is the opposite hormone, which reduces the appetite. So we investigated whether this pathway is also changed in the brain with the diet-induced obesity in adult animals. So similar diet regime, we fed the rabbits with a high-fat diet or a normal diet for 3 weeks, and we gave these drugs directly into the ventromedial and the dorsomedial hypothalamus. Now, these graphs look a little bit complicated, but it's pretty straightforward. The top one shows the blood pressure, heart rate, and the renal sympathetic nerve activity in the bottom. The red shows the high-fat diet feeding, and this is a dose-dependent time course graph. And the blue represents the control diet, and the gray ones show the ringers, which is the vehicle when we gave these drugs to the brain. So as you can see, when the leptin antagonist was injected in the 3-week high-fat diet fed animals, there was a reduction in blood pressure in a dose-dependent manner, meaning when we gave more stronger dose of the leptin antagonist, there was reduction in blood pressure and also heart rate as well as the renal sympathetic nerve activity. And on the right-hand side, this bar graph shows the average change from the control period. So we know in the ventromedial hypothalamus, leptin antagonist reduces the blood pressure, heart rate, and sympathetic nerve activity. Alternatively, when the alpha MSH was given, so when we gave more of this alpha MSH to stimulate this pathway, there was marked increase in blood pressure and renal sympathetic nerve activity in the high-fat diet animals, but no change in the control diet apart from the heart rate. So we know that the alpha MSH pathway is also altered in these animals. Now, when we blocked this with the SHU9119, the results were similar to the leptin antagonist, where we saw a marked decrease in the blood pressure, heart rate, and sympathetic nerve activity. Surprisingly, when we gave the MPY injection, again, this is the stronger dose of the opposite hormone. So we were expecting when this was given, the blood pressure and sympathetic nerve activity might reduce. However, there was no change in the blood pressure, but a small increase in the renal sympathetic nerve activity, indicating that the MPY signaling pathway is not activated at this stage. And when we also gave it to the dorsomedial hypothalamus, again, there was no change in any of the measurements. Now, you might ask, how do we know whether we hit the VMH or the DMH in the conscious animals? And this was investigated after the last experiment. We injected a dye to see the injection site, and as you can see, this one clearly hit the dorsomedial hypothalamus, and this one is a ventromedial hypothalamus. And we only used the data that shows that it hit the target area. So with this project, we discovered that the ventromedial hypothalamus has an important role in the leptin-mediated renal sympathoexcitatory pathway, and the response to alpha MSH and SHU-9119 also show that the leptin and melanocortin signaling pathway resembles each other. Hence, we concluded that both alpha MSH is likely to use the same pressor and the sympathoexcitatory pathway in the VMH, resulting in obesity-related hypertension. So, combining the ICV study with the VMH study together, what we know from these exciting findings are that we know that the main cause of the neurogenic hypertension in diet-induced obesity in adulthood is due to the changes in the leptin signaling pathway, as well as the downstream, which is the POMC signaling pathway. Oopsie. So, we know that, and we also know where this is occurring. So this is happening in the ventromedial hypothalamus, and we have shown that this is the origin of the changes in blood pressure and sympathetic nerve activity in obesity-related neurogenic hypertension. Okay. Now, these interesting findings have led to my next project, where we have a strong interest in the programming effect of obesity and related hypertension. We're now focusing on the developmental origins of obesity-related hypertension. And we strongly think the leptin and insulin, as well as the melanocortin signaling pathway, is a strong contributor in the changes in this developmental programming of hypertension. So in simple terms, when mothers are obese during pregnancy, this can affect the development of the offspring's autonomic nervous system, or the brain, and this can be a starting point of these offspring developing more severe conditions of obesity and hypertension later in life. So, our main hypothesis is that exposure to the excessive adiposity due to overnutrition in utero and during lactation, the offspring will experience altered development in the brain, in particular with the key receptors in the appetite hormones. And this includes the neurotrophic action in the hypothalamus. Now, these overlying conditions will lead to changes such as reduced metabolic action and also increase in sympathetic nerve activity, making these offspring more prone to develop obesity and hypertension when they are exposed to an obesogenic environment later in life. And we think this, again, is due to the action of leptin and insulin signaling pathway in the brain. So the aim of the developmental programming study was to investigate the effect of maternal obesity on the changes in the cardiovascular parameters and renal sympathetic nerve activity in the offspring. And we wanted to determine the mechanism of this programming effect of hypertension and the sympathoexcitation. So, I know you guys study a lot of nutrients in your study, but in this particular study, we are mainly focusing on the high fat. So there are a lot of foods in Australia and also around the world, as well as in Korea, with the high fat content. And this study focused on what does that high fat content ingestion from mothers due to the offspring when they are in adulthood. So we fed the mothers with the high-fat diet for 3 weeks prior mating, during pregnancy and lactation. Now, once the offsprings are weaned, meaning when they start to eat solid food by themselves, they were only fed with a controlled diet throughout their growth. So the period that these animals, the offsprings, were exposed to the high-fat diet is only when they were inside the mother, so during pregnancy and lactation period. Now, all the offspring were implanted with this bilateral cannula, so we could target both ICV as well as the areas that we targeted in the adulthood study. And they also received the renal electrode to measure the renal sympathetic nerve activity. So similar to adulthood study, we injected leptin antagonist, alpha MSH, SHU9119, as well as the insulin antagonist. And we also investigated the messenger RNA expression in the brain at the end of the experiment. So we looked at the mRNA expression of leptin receptor, the melanocortin 3 and 4 receptors, and also the BDNF, which is a brain-derived neurotropic factor, and MAP2 and ERK receptors. And these neurons are what we think is driving these changes in the physiological level. So we wanted to see whether that is true. Now, to compare with our adulthood study, we also had a group which were only fed with a high-fat diet for 10 days in adulthood. So this is the green one is to compare with the adulthood obesity to the developmental obesity. So after a challenging period of time, we can now show you our exciting results where we could see the programming effect of maternal obesity on the offspring health, the program effect of maternal high-fat diet consumption in the offspring. So I asked you guys to focus on the red color throughout the presentation from here, which shows you the effects in the offspring of maternal high-fat diet-fed animals. So in these groups, there was marked increase in blood pressure, heart rate, and renal sympathetic nerve activity in adulthood. Now, and again, I have to emphasize that these animals in red were only exposed to the high-fat diet during pregnancy and lactation. So clearly there is an effect from the maternal side to the offspring in adulthood. Now, another exciting finding was that in the maternal high-fat diet group, there was an increase in fat. So they got fat just because their mothers were fat, or they were eating a high-fat nutrient when they were having the babies. But the plasma contents show there was no difference in plasma leptin or glucose in these offspring. Now, let's go with the ICV again. So on the left-hand side, it showed the ICV injection. Again, ICV injection, the drug goes to everywhere of the brain. So when we gave the drug in the ICV injection, there was a reduction in blood pressure, heart rate in the maternal high-fat diet only. There was a trend of reduction in the renal sympathetic nerve activity, but we didn't see a significant change. But there was a marked reduction in the blood pressure and heart rate when leptin antagonist was given ICV. Now, when we directly targeted the area of the ventromedial hypothalamus, there was not much change, showing that we were actually surprised when we didn't see any change with the leptin antagonist. But we clearly saw with the ICV injection, there is a change due to the maternal obesity in the brain of the offspring. Now, a hypothesis that we didn't see any change in the VMH was that as we saw in the adulthood study, this delayed action of leptin could be kicking in here. Hence, we didn't see any changes in these animals. Now, when the alpha MSH was given, there was really exciting findings where both ICV and VMH showed marked increase in heart rate and renal sympathetic nerve activity in the maternal high-fat diet animals, as well as a blood pressure in the VMH injection. So, clearly showing that these results in the offspring is similar to the adulthood study. And this is clearly a programming effect. Again, this is all due to the mother's high-fat diet causing the changes in the blood pressure, heart rate, and renal sympathetic nerve activity in the offspring in adulthood. Now, importantly, these animals had no exposure to the high-fat diet after they were born or after they start eating solid food by themselves. SHIELD 9119 also showed a significant reduction in blood pressure, heart rate, and sympathetic nerve activity in ICV injection, and also a significant trend in the VMH injected maternal high-fat diet group. Again, showing that this may be caused due to a delayed action of the high-fat content in the offspring. But these results show that the sympathoinhibition by the SHIELD 9119 and the excitation by the alpha MSH, as we've seen in the previous slide, these only occurred in the maternal high-fat diet-fed animals, offspring of maternal high-fat diet-fed animals, showing that this is clearly a maternal high-fat diet programming effect in the blood pressure and the cardiovascular parameters. And again, this is a programming effect. Now, insulin antagonists surprisingly didn't do anything. So we think the action of leptin is masking the effect of the insulin. Because the effect of leptin is so strong, we can't detect the other action of the insulin. But we are still investigating whether this is true. Now, when we looked at the hypothalamic mRNA expression using a PCR analysis, I'm pretty sure you guys are very familiar with PCR now because we do it every time with the COVID, checking for the COVID infection. We saw a marked increase in the leptin receptor, BDNF, SOX3, melanocortin-4, ERK, MAP2 kinase, and PI3K, all involved in the changes in the sympathetic nerve activity. So this, we think the changes that we observed with the drug injection are due to the alteration of these precursors of the signaling pathway in the hypothalamus, because these neurons or these genes are the ancestors of the parts that drive the changes in the leptin signaling, melanocortin signaling, as well as the insulin signaling pathway. And we think, and we strongly suggest, this is due to the programming effect of maternal high-fat diet. So with our results and observations in this maternal obesity and offspring programming study, we concluded that the greater central leptin signaling and the greater sensitivity of the receptors are driving the increase in the blood pressure in the programmed offspring. Now, this is mainly due to the maternal programming effect, as it altered the central leptin melanocortin signaling pathway in the hypothalamus. So, in summary, we have shown that maternal obesity programs altered leptin and melanocortin signaling pathway in the offspring, in utero, and during early development, which initiated the neuronal plasticity. And both leptin and alpha MSH is likely the main cause of changes in blood pressure and sympathetic nerve activity in the programmed offspring. Hence, blocking and preventing this neuronal plasticity will lead to improved cardiovascular outcome in obesity-related hypertension. With the outcome from our studies, we have a strong opinion on the mechanism of programming of obesity-related hypertension is due to the programming of localized neuronal plasticity. So we suggest that during development, leptin enters the fetus across the placenta, and it promotes the activation of POMC neuron in both arcuate and the ventromedial hypothalamus to release alpha MSH and activates this melanocortin signaling pathway. And over time, this strengthens the sympathetic transmission, leading to amplified leptin and melanocortin signaling, despite relatively normal plasma leptin in adulthood. And this eventually causes the exaggeration or exaggerate the sympathetic excitatory effect of leptin and hypertension that we observed in the programmed obesity. Importantly, there are studies indicates that increase in melanocortin signaling is closely linked to the important neurotrophic factors such as the MEK, ERK, and the BDNF. So we want to test this theory by investigating the neurotrophic factors which are responsible for hypersensitivity of the receptor. So, our most latest plan is investigating the changes in this signaling pathway in accordance to the chronic leptin and melanocortin responses. So we have preliminary data showing that the BDNF expression is increased in the offspring. Again, BDNF is brain-derived neurotrophic factor, which is, we think, the main reason why there is hyperactivity in the brain when leptin or alpha MSH is introduced to the brain. So we have preliminary data showing the BDNF is increased in the VMH, and when this was blocked using a MEK inhibitor, the obesity-related hypertension was abolished. So to investigate the mechanism underlying the programming of hypertension in the in offspring of obese mothers who fed with a high-fat diet, we will investigate whether the increase in sympathetic nerve activity is driven by the programming effect of BDNF-dependent amplification of leptin, melanocortin, pre-sympathetic pathway in the VMH. I know it sounds really long and complicated, but it's pretty simple. So we're looking at whether the high-fat diet in mothers are affecting the development of the brain-derived neurotrophic factor in the offspring, and whether that change is making this whole hypertension occurring in the offspring by the leptin and melanocortin signaling pathway. So we're narrowing it down a little bit more to the specific part of the brain driving these changes. So to do that, we will use, again, the rabbit offspring model, as well as this time we're using a viral knockout of BDNF in the rodent model. So in the mouse offspring, they are genetically modified to have a knockout in the BDNF. Not all BDNF that will kill the animal, but 70% of BDNF is reduced, and we will see whether when we feed the high-fat diet to those mothers, what happens to the offspring. When we get that result, we will clearly know that the BDNF is the main cause of all these changes. In addition, I think I focused way too much on obesity only, but we are also looking at the alternative approach in diabetes induced by obesity. So we are using a citrus bioflavonoids. This is like I don't know what this in Korean. It's like citrus. 신 음식이라고 하나요? The supplement of the citrus bioflavonoid as a therapeutic approach to the glycemic control and vascular health in obesity and type 2 diabetes. And we think the current treatment for type 2 diabetes in Australia is using a specific drug which called the gliptin, but this has a nasty side effect. But the citrus bioflavonoids is the nutrient. I think you guys are familiar with this because many Mediterranean food or Mediterranean diet uses a lot of citrus in their food. And studies have shown that by intaking a lot of citrus bioflavonoids, in our case, if we use a citrus bioflavonoid supplement, this will help controlling the blood glucose level and eventually become an alternative approach to treat type 2 diabetes and also the pre-diabetes, which is common side effect of obesity. So this will be updated soon in the future. So finally, I would like to acknowledge my collaborators and friends who helped us completing these studies, and also the funding body, which is the Australian government and the National Health and Medical Research Council of Australia. And yeah, thank you very much for listening. And hopefully, if you have any questions, send me an email. And I just would like to finish off a very popular image that you guys all know, our campus in Sydney University, and also the landmark, the Opera House and the Harbour Bridge. Thank you very much for your attention. 감사합니다.